Hello and welcome to this video on codominance. We are exploring a few different inheritance patterns when it comes to genetics. Previously, you took a look at a dominant-recessive relationship among alleles, different versions of the same gene. And here we're going to take a look at an instance where one trait doesn't mask or cover up the other trait. So let's get into it here, talking about codominance. I've included a slide that has a vocabulary recap for you. I'm not going to take time to go through each of these terms, but it's really important that if you don't already know what these terms are, I'm going to be using them in this video. They're going to be very important for working the problems at the end of this video, as well as the ones that are assigned to you in class, and then again for the test. They're the basics as far as communicating these genetics ideas for us. So make sure we know what a gene is, what alleles are, the difference between a genotype and a phenotype, what the difference between homozygous and heterozygous genotypes are as well. So review those real quick if you need to. Jot them down, have them handy so that you can refer to them at any point during the video if you don't already have them down, and or have your study guide handy. You can fill them out on your study guide. Be a great thing to do. All right, let's just define codominance, and we're going to keep it really simple here. In codominant inheritance, both alleles for a given gene are expressed. In simple dominant recessive, it was the dominant trait that we saw if dominant and recessive were present. So we're talking about a heterozygous individual where they have two different versions, two different alleles for the gene. So another way for us to describe codominance is in a heterozygous individual, both phenotypes appear. Remember, again, phenotypes are the physical manifestation or observable a characteristic of the gene. So both of those traits are expressed. So how can we figure this out or picture this? Think about pizzas. Let's pretend pizzas are living things, organisms, and they have a gene for toppings. And there are multiple alleles for topping, the topping gene. Let's say you can have a pepperoni allele, you have a sausage allele, you have green onion allele. So we've got tons and tons and tons of different alleles for the topping gene. Let's explore two of those. Let's say that pepperoni and sausage are codominant traits. So if I have a genotype down here, indicated by big P, what would the phenotype be? So we have two copies of the pepperoni allele. Well, because there's nothing else present, we're going to see a pepperoni pizza. That's what we observe. That's the phenotype. What about a homozygous sausage pizza? Well, you're going to see the sausage phenotype displayed. But with codominance, what if we have one copy of the pepperoni and we have one copy of the sausage? Well, like we said, both traits get expressed. We're going to observe both phenotypes, which means we've got a sausage and pepperoni pizza. Yay, excellent. And now that's making me hungry. So we see both of the traits observed rather than one covering the other up. So some examples. Flowers. Flower petal colors can follow a codominant pattern. Rhododendrons in particular. If we have a red individual, we can look down here at this, Punnett square. Red would be big R, big R. White would be big W, big W. So a red flower would have two big R's. A white flower would have two big W's. But what happens when you have big R, big W? Well, you're going to see both traits. So in the picture here, which is covered up a little bit by my video, sorry about that, we've got both white and red present in the flowers. This nomenclature here too is a little bit different. We're going to practice this at the end of the video, but it's instead of the same letter representing the gene, right, capital lowercase to show dominant recessive, here we're using two different letters, both capital, to show that both are going to be expressed. Okay, so this happens a lot in livestock. Chickens. We can have a chicken that has a big B, big B. We can have a chicken that has big W, big W. Big B would be a all black phenotype. Big W is an all white phenotype. But if we have big B, big W, we're going to express both black and white as part of this. So we wind up with a speckled black and white chicken here. So codominant, both traits are displayed. Same thing can happen with cows and horses with this coat pattern called roan. And what happens there is some of the fur is red, some of the fur is white. It's not a blending of the two. That's a different type of inheritance. But instead, we see both white and red being expressed at the same time, codominant. As far as humans are concerned, there are a couple of examples that we could take a look at. You're going to see one of these later on with Miss Ripley is going to do a video on blood types where we'll see some codominance. But also dealing with blood, sickle cell anemia is also an example of a codominant relationship. And sickle cell anemia, if we haven't talked about this already at any point in your classes, the blood cells in sickle cell anemia take on an elongated, narrow crescent or sickle shape. Some students describe it as a banana shape. A regular blood cell typically is round. It has like a little bit of a divot in the middle, looks a bit like a donut. So these sickle cells have a different shape to them. So if you have the gene for making the normal type of blood cell and the gene for making sickle cells, well, you're going to express both the normal and the sickle cell version. So you're going to have combination in your blood of sickle cells and regular cells. And you can see that in this microscope image here. We've got some normal red blood cells. We've got some sickle cells. So this is a codominant individual. They have one copy of the sickle cell gene, one copy of the regular cell gene, and we see both traits expressed. If you look at this chart here, there are, as far as sickle cell anemia is concerned, there are different ways of interpreting this here. We're talking about, as far as the phenotype of the blood cell shape, that pattern is codominant. If we're talking about whether or not you have anemia or whether you're malaria resistant, that one's actually a dominant recessive relationship. So genetics, like we've alluded to already, is really, really complicated, far more complicated than what we get into here in our class. But I want you to be aware of it and also hopefully inspire you to take a look at more examples then later on. Practice problem time. We've got 3 practice problems. I'm going to go through them here. You're welcome to, once you feel confident in your ability to complete these problems, you can go off, you can work on the practice set on your own. You can come back to this if you find that you're running into trouble. I also would recommend, if you've watched all 3 examples here and you're still in need of more assistance, 1, ask one of your teachers. We're happy to help. 2, YouTube is full of lots of videos that help explain and walk you through these types of problems. So let's try this out. I'm going to pause at certain points here and ask you to help me out with figuring this out here. I like to make a little bit of an inventory at the start of this. So I'll spend more time on this first problem showing you that setup, and then we'll kind of go faster in the future ones. So I've got some key terms here. Homozygous red fur for the horse and a heterozygous roan horse. So I need to figure out what are the genotypes of the parents. Remember, again, genotypes are the allele combinations, right? Are you what letters do you have for the genes? What versions of the alleles? So homozygous, homo means same. So homozygous is 2 of the same. So if it's homozygous red fur, I've also got this noted over here. Red fur is being represented by the capital letter R. So homozygous means 2 of the same. So one of the parents is big R, big R. And the other one, hetero, means other or different. So we have 2 different alleles. And we're also told it's roan. So we know that we've got a codominant situation where we're getting both phenotypes displayed. So I know that this individual is going to be big R, big W. There we go. That being the case, I need to set up a Punnett square. So I'm drawing my square here. I am not going to be concerned on this problem here as to which parent goes on the side, which parent goes on the top. A bit irrelevant. So you can set, if I pause here and let you work the problem out, and then you can play and show it back. If you set it up differently than me, like you put the on the side rather than the top or vice versa, totally fine as long as the results are the same. We should get the same results. So I'll pause here for just a moment to let you set up this problem and answer these questions. What are the possible genotypes and phenotypes of the offspring? Okay, so I am just going to put big R up here at the top. Like I said, maybe you put it on the side, it doesn't matter. Big R, big W. And the way that this works, right, I carry this R in, I carry this R over, and I'm going to wind up with big R, big R. Same deal here, I carry this R down, I carry this R over again, so I've got big R, big R. This R comes down here, the W comes over, RW, RW. Excellent, so I've got the Punnett square. Now I need to figure out what the possible genotypes and phenotypes are. I'm going to make a little chart here. Geno, pheno, and I'm going to just kind of associate which one goes with which. So we have big R as a possibility. Big R. And because this is codominant, and I have 2 copies of the red allele, I'm going to see red. This is like that pepperoni pizza or the sausage pizza, right? If we're just coding for pepperoni or sausage, that's all we're going to see on there. So this one is going to be red as the phenotype. And if we want to throw the percentage on here, 2 of the 4, or 1/2, or 50%, I'm running out of room here. All of those mean the same thing, right? 0.5. You could say that too. Half the offspring would likely be red. And now I've got the genotype RW, and RW is going to be a situation where red is produced and white is produced. So this is going to be roan as our phenotype. Again, that's the other half, so I'll just put 0.5 down here to show that it doesn't matter how you present it. I'm not going to be picky. I don't think any of the other teachers are picky about how you list the probability, provided it's some sort of equivalent of what the right answer is. Okay, that's our first problem. Here we go. Let's do next one. Is this going to let me? Maybe I need to erase all of this before I move on. Hopefully not. Oh no, I do have to erase it. Give me a moment here. Oh no, this is going to take forever. Is it like a whiteboard where you can draw a circle and click? Nope. What about wipe screen clear? That's what we wanted. Perfect. I'm learning. All right. Codominant practice. Here we've got rhododendrons. We got some flower issues going on here. Heterozygous. Again, I'm just going to highlight my terms. See if I can make the pen a little bit easier to see. Pointer. Oh. Looks like that's what it's going to be stuck at. And a white rhododendron. R is red, big W is white. So once again, I'm going to try and figure out what the genotypes of the parents are. See if you can figure out what a heterozygous rhododendron would be. See if you can figure out what a white rhododendron's genotype would be. So heterozygous means 2 different alleles, and the only 2 alleles I've got in this problem are R and W, so I must have a big R, big W. So that's the hybrid version, right? We're going to have red and white displayed on the flower petals. That would be the phenotype. And then white, the only way to get white is with 2 white alleles. So I need W, W. So now I set up my Punnett square. And again, depends on however you want to set it up here. We haven't said who goes where, but I'm going to put WW, RW. Fill this out. This R comes down, this W comes over. I'm just keeping it alphabetical here, right? R comes before W. In this square, this W comes down, this W comes over. So we have WW. Down here, we have another RW, and here is WW again. Okay, time to predict the possible genotypes and possible phenotypes that go along with them. Let's add in here a probability column too. Prob. All right, genotypes. What are the possible genotypes? RW and WW. We didn't get any other combinations, right? The only other option that we don't see here would be big R, big R. But none of the offspring can be big R, big R. So no all red offspring. RW, phenotype is going to be both colors displayed. Why is this, there we go. Woo! Sorry, we've got computers freaking out just a little bit. Okay, so we're gonna have both colors. It's gonna be red and white. And then probability, well, we have 1, 2 out of 4. So that's 50%, 1/2, 0.5, 2 out of 4. All of them mean the same thing. And then WW, that phenotype is white. It's an all white phenotype, and the other 50%. Excellent. One more practice problem, and then you can see if you can get this going at home. If I do this, there we go, clear all. See, I can learn. And we have our last problem here, which is 2 individuals are both heterozygous for sickle cell anemia, and they're expecting a child. What are the possible genotypes and phenotypes? And we've got our code here. Big S is sickle cell, big N is normal cell. And we're talking about the cell shape as our phenotype. And then we also want to know what are the odds that they'll have a child with sickle cell anemia. I don't know where the other L went. There should be another L there. Okay, I'll pause here. I'm going to let you set up your problem, and then we'll come back and we'll work through it and show you the answers. Okay, so genotypes of the parents. Genotype, they're both heterozygous. Once again, hetero means difference. We have 2 different alleles. The 2 different alleles we're looking at here are S and N. So both parents are going to be big S, big N. I'm going to draw my Punnett square. Big S, big N, big S, big N. And fill it in, right? Big S, big S, big S, N, big S, N, and N, N. I didn't follow my alphabetical rule here, I'm sorry. Well, we'll try to be more consistent in the future. So what are the possible genotypes? What are the phenotypes? And what are the odds? So we have 3 different genotypes: big S, big S, big S, N, and big N, big N. Big S, this is going to be all sickle cell as far as the shape is concerned, all sickle. Odds, how many of these are big S, big S? We've got 1 out of the 4. So this is 1/4, 25%, whoops, 25% or 0.25. Once again, doesn't matter what you say, as long as it's one of those. And big S, big N. We're going to have both cell shapes, right? We're referring to normal and sickle shapes. And there are 2 out of the 4 there. We could simplify that to 1/2. And then there's big N, and this would be all normal shape. This all normal. Sorry, handwriting got out of the way from me there. And that is 1 out of 4 again. Final question, what are the odds they will have a child with sickle cell? Well, there is only 1 where we've got both of these sickle cell genes here. If the question, you know what, maybe this is a poorly worded question, because does it mean that they only have sickle cells? If we're talking about only sickle cells, then it's 1/4. If we're talking about any sickle cells, well, then it's going to be 3/4, because all of these offspring produce some sickle cells with their phenotype. So I should have maybe written that question a little more clearly. But that is codominance. And like I said, if you need more practice, come back, rewatch the video, look up some videos on YouTube, swing by, ask some questions for your teacher, stop in during ET, all of that good stuff. Hope this helps, and good luck.